Hi, it's really cool, but also a little bit intimidating to be the first talk to follow the session about forest ecology, management and bioindicators, because I am part of the field that is helping to develop the tools that people can use in forest ecology, management and bioindicators. So I hope I can capture your interest with my talk about one specific part of the tool development. But I guess it's really important to make this step between the tools that we're developing and implementing them in forest ecology and management. So I hope that while I'm talking about this one research question that I'm doing to develop the tools, it might showcase the things we can do with stable isotopes and also like our approach to them, so that it shows how flexible we are when we're developing them as a tool to tailor them as a tool for different research questions. So in this talk, I'm looking at leaf water heavy isotope concentrations in relation to ecosystem processes in boreal forests. Sorry, it was not. Oh yeah, here, sorry. Yeah, so why leaf water stable isotopes? Why don't you want to look at tree rings? Well, if we start on the left here, we've got evaporation conditions in the forest and source water variability that's impacting the leaf water isotope variability. And don't let the D confuse you there. You know, H2O is water, H, that's the D, and then O, that's 18O, and, oh, this isn't. Sorry, I have to click here. Yes, so 18O is the ratio of the heavy oxygen isotope compared to the lighter oxygen isotope relative to a standard we use. And yeah, so D deuterium is the ratio of heavy isotope of the hydrogen compared to the lighter isotope of hydrogen. So we've got these isotopes in the leaf water. Why do we want to know about them? Well, those signals that are stored in leaf water are transferred in biological processes in the trees, eventually to long-term bioindicators. So the signals that we're measuring in tree rings or in any animal tissues, if the animals have been eating plants, at least the plant signals inside the animal tissues, or other long-term bioindicators, that is coming from the processes that formed the signal. And for what I am looking at, which is the water signal, that's the signal in leaf water. So that's why it's so important for us to look at this origin signal in leaf water. And leaf water isotopes have been related in the past to individual environmental variables like relative humidity, vapor pressure deficit, and source water isotopic variability. But something that's lost when we're interpreting these stable isotopes is that actually when we walk in an ecosystem, all of these different factors are interconnected. You haven't just got relative humidity changing in a laboratory experiment. There's VPD change at the same time and power change. And all of these different environmental factors are also linked by ecosystem processes. So in this study, we explored the relationship between leaf water heavy isotope enrichment and one ecosystem process, evapotranspiration in boreal forests. Here there's a link between many different environmental factors, relative humidity, temperature, power, soil water content, and the ecosystem process evapotranspiration. And this relationship between the multiple environmental factors and evapotranspiration is well known. You can model evapotranspiration from those factors. And then we also know that leaf water isotopes are related to individual environmental factors, but they haven't so much been looked at collectively. So that's the first research question in this study, which is to see, like, when they're combined, how they are representing leaf water isotope variability. And then when it comes to evapotranspiration and leaf water isotope variability, there's some uncertainty when it comes to this pathway that I illustrate, because people can really go into the maths, they can model evaporation and transpiration, but that relies on models which still require improvement. So here we're kind of getting back to the basic environmental factors and how they're related to evapotranspiration and leaf water isotope variability. So that's the second research question, how evapotranspiration relates to leaf water isotope variability. Yes, so for the methods, we used 2 study sites in Finland using an ERC project, boreal, and it was during the 2019 growth season. And one sampling site, the lower sampling site, was Hyyitälä in the southern boreal zone, and then the northern boreal site was Värriö, which is at the northern tree line. And then there was an isotopic survey where there were soil water samples, tree organ samples, and water vapor taken. And there were 3 to 5 trees sampled per sampling day. Both of these sites were ICOS sites, so that was really helpful because there was continuous meteorological and eddy covariance data available for this sampling year. So looking at the results, like I have just selected 2 key variables in the environment to show the temporal trends of, but we were looking at many. But between the 2 sites, generally Värriö, the more northern site, had a shorter growth season, and both Hyyitälä and Värriö exhibited highly variable vapor pressure deficit. In case you don't know what vapor pressure deficit is, it's the amount of vapor in the air that it could, oh gosh, I'm going to get mixed up with what could not and could not hold, but it's how much is lacking compared to what it could hold. And then, so that's the top graph, and the below graph is soil water content. I got that one there because that was one noticeable difference between the 2 sites. Värriö, in the more northern site, the soil was generally wetter than at Hyyitälä. I just said that. And then when we come to the many different relationships between environmental factors and isotopes, we've got this one graph here to visualize how they're related to isotopes. But really, you know, for the stats, we did some regression style analyses, generalized linear mixed models, but rather than show many correlations rowed up, we've got this redundancy analysis to show how these different factors are interacting. So if you look on the x-axis, that's redundancy analysis dimension 1, which is explaining 74% of the variability in the data. And then on the y-axis is like this second dimension, which is explaining only 5.4%. So when you're looking at this graph, the things that are changing from left to right are much more influential to the isotope variability than what's happening on the y-axis. And also, when you look at these different arrows, the longer the arrow is, the greater the effect it's having on the isotope variability. So you can see that some of the biggest drivers in the stable isotope variability are this negative relationship between relative humidity and VPD and PAR. And it makes sense with VPD because it's calculated from relative humidity, but it's still a really commonly used parameter. And it's also calculated from temperature. So we can see here actually that relative humidity is like at these sites driving VPD more than temperature. And I think this is also really a valuable graph because often people might infer like relative humidity on its own relating to these isotopes, but you can actually see like if you did that at these sites, well, how can you ignore VPD and PAR because they are correlated? Can you really just say it's relative humidity? Yeah, and something that's interesting on isotope level is that the source water isotope variability was not as important to the isotopes in the leaf water compared to the soil water content. That's interesting because, you know, some people can trace the precipitation and, well, the soil water content was more important here at these 2 sites. And so the final results were showing relationships between the isotopes and evapotranspiration. And we found that evapotranspiration was significantly correlated to leaf water isotopes, but it's also showing some really interesting trends about context dependency at these sites. Firstly, one of the water isotopes was more strongly related to evapotranspiration than the other isotope. And also at one site, the more northern site, evapotranspiration was having extremely close relationship to oxygen, leaf water oxygen 18 concentration. So if you look on the bottom graph, you can see like this R squared equals 0.95 relationship. And so that's quite an incredibly strong trend going on there. So to conclude, leaf water isotopes can be strongly related to ecosystem processes such as evapotranspiration. And this is because evapotranspiration is related to multiple interacting environmental variables. And together, those variables can be strong descriptors of leaf water isotopes. But as you can see by the site differences we observed, there's still an opportunity to explore site differences further to know why at one site there's a stronger relationship between evapotranspiration and leaf water stable isotopes than at another site. Thank you for listening.
Thanks, Charlotte. There is the question in the room.
Yeah, those are some interesting data. Do you think so? Because trees are really reactive to extreme events that happen over short duration, right? So do you think there's a potential to draw relationship between these leaf level measurements and then the integrated isotopic measurement that occurs in a tree ring?
Yeah, like that's definitely what people do. And it's also what my project is doing. I'm reporting results from the leaf water, but next we will take these results to the sugars and then to tree rings. So yeah.
Yeah, okay, cool.
Yeah, Clemence.
Yeah, thank you for your presentation. Thank you for explaining LTA graph. I had a question, but I think I saw the answer on the last slide. What is ICOS? Because you said it's an ICOS site, and
yeah, it's a network. It's called the Integrated Carbon Observation System.
Okay. Perfect. Thank you.